Good afternoon, my name is Kartik Subramanian. I am also a PhD student in Dr. Tyson's lab. Now I've been working on my project, which you can see the title here, is the spatial-temporal model of the cell cycle of an organism. That's a pretty heavy title, but I've been working on this for about 3.5 years, and I find plenty of things interesting about it. But due to the shortage of time, I'll just tell you one thing that I find really interesting is the notion of an asymmetric cell cycle. So what is that? We know what a general cell cycle is. The cells grow over time. When they're big enough, they divide, and they give two daughter cells which are identical to each other. They have the same DNA and the same composition of proteins, right? If you want to study the cell cycle, the way we do it is to understand what the underlying regulatory network is, convert that information into the language of differential equations, and then we solve those equations to get time courses and bifurcation diagrams, and these are tools which hopefully give you some insight about the underlying physiology of the cell. But like I mentioned, I work on something called as asymmetric cell cycle. So what is that? This is a picture of the Colobacter cell cycle. And what you'll find here in the cell cycle is that the two ends of the cell look different. When the cell divides, they give rise to two different types of cells. In the sense, the protein composition of the cells are different. So this is an asymmetric cell cycle. And the reason this asymmetric cell cycle is generated is because the sort of biochemical reactions that happen at different points on the cell is different. So you can appreciate why we need to have a spatio-temporal model. We need to have a model which not only captures dynamics in time, but also in space. So I've been lucky enough to work with Dr. Tyson as well as Dr. Mark Fall from the Department of Mechanical Engineering and Dr. Yankov from the Department of Computer Science. And together we have come up with some interesting results talking about why this asymmetric cell cycle happens. Very quickly, I'll just give you a sample plot of mine. This is a spatial-temporal plot of the space on the y-axis and time on the x-axis, and this tells you how the protein concentration changes inside the cell over that time. But the intensity of the color plots tell you the concentrations. So if you're interested, you can come by and look at my poster and we can talk more about this pretty interesting phenomenon. Thank you.